And we're live. Dr. David Ingram, please take it away.
Good afternoon. Welcome to the Science Cafe, sponsored by Sigma Xi, the Scientific Honor Society, and the Office of the Vice President for Research. I'm David Ingram, the Vice President of the local chapter of Sigma Xi and the Chair of the Department of Physics and Astronomy. This week's cafe, which will be the last one for the fall, will be given by Professor Nathan Weyand of Biological Sciences. He'll be talking about microbial colonies and protected blood. Before he begins, though, just a reminder, the Science Cafe information can be found at ohio.edu/Science Cafe. And we really would like you to, in the spirit of the Science Cafes, provide some questions for our speaker as the talk moves along. So please put those questions in the chat, and these will be asked by Rox during the talk. Now over to Nathan.
Thank you. So I'm hoping that my presentation today will help you have an idea of what it's like to be a bacteriologist. When people ask me what I do for my profession, I'll often say I am a bacteriologist or a cellular microbiologist. So I'm hoping that today's presentation and discussion with you will help you understand those terms a little better. So I'm going to share my screen now and start showing you some slides. Okay, this first slide has the title of my presentation on it, but these are pictures of bacterial colonies, and so I'm going to help you understand what a colony is today and show you some pretty pictures that I hope that you enjoy as much as I do. So the term microbiology has this micro in it that's underlined, okay? And so we're dealing with biology at a small scale, a micron scale, and so this is 1/1,000,000th of a meter, okay? And what this picture is that I'm showing you is a scanning electron micrograph of the point of a pin at different magnifications. And so as you go from left to right, you can see these orange cells appear, and these are bacteria. So they're rod shaped. This is a very common shape for bacteria. A general definition of bacteria could be a single cell that lacks a membrane-limited nucleus. Okay, and bacteria have lots of different shapes. Some of the bacteria I study in my lab are shaped like this. And this is called a diplococcus. So many bacteria are spherical. And so a spherical shaped bacteria is a coccus. And if you have two spheres together like this, it's called a diplococcus. I want to tell you about some of the tools we use in my lab. So you have a kind of an understanding of some of the things we do. This is a picture of pipettes, and your hand holds the blue portion of these instruments, and there's plastic tips on the ends of these pipettes, and I've drawn up liquid into the tips so you can get an idea of the different volumes that we might work with. So the pipette on the right is showing a 1 milliliter volume, and then we progress from right to left to a 2 microliter volume. We use these a lot in the laboratory to transfer bacterial suspensions between tubes or to make solutions for some of our experiments. So these are for small volume manipulation. We also use larger pipettes, shown here, for larger volumes. So the top pipette here holds 50 milliliters of liquid, and 25, 10, and 5. And your hand holds this, and you draw liquid up by pushing these blue buttons or eject it. One thing we use a lot in a microbiology lab are Petri dishes. There was a German bacteriologist named Petri, and he developed this dish as he worked in Robert Koch's laboratory to help growing bacteria. And so we order these pre-sterilized in bags. And so on the left, this bag here, plastic bag, holds 25 sterile Petri dishes in it, and each box has 500 dishes in it. On the right here, we're looking at some empty Petri dishes, and then some that have solid growth media in here. And so we can inoculate bacteria onto these solid surfaces, and bacteria will grow. All right, this slide, I apologize for the sternness on my face here, but this slide just shows you some of the personal protective equipment that I wear in the laboratory, and my students also do. This was taken before the pandemic, so I'm not wearing a mask. We all are wearing masks now when we work in the laboratory. But you'll notice I'm wearing safety glasses. So one of the organisms we work with is Neisseria gonorrhoea, and this, if it gets in your eye, can cause a very severe eye infection called conjunctivitis. So we all wear eye protection to avoid that. We wear lab coats and gloves to try to minimize contact with the bacteria. And you'll notice I have a swab in my hand. This is another common thing we use in the lab to remove bacteria from the surface of the agar in the Petri dish. So I'm going to show you a movie now, and I'll talk over it just to explain what we're doing, what I'm doing. So I'm taking some sample out of a cryo vial. This is a vial we store in a freezer, and it's a sample from a throat swab. And the material's frozen. I'm scraping some out with a pipette tip, and I'm going to put it on the solid surface of the agar in the Petri dish. Okay, so and then I put the vial back in a cooler to keep the sample frozen. So we try to minimize freeze thaws when we're processing our samples. Now I'm getting what's called an inoculation loop out of a bag. These are sterile plastic disposable loops, and I'm going to spread the sample out on the plate. Now I'm going to throw this loop away, and I'm going to get a new one out, and I'm going to go through this process we call streaking again. And this process helps dilute the sample so that the bacteria land where we have one bacterial cell land on the agar. And then we put the plate in an incubator, and we let that grow. And where you have one bacterial cell on the agar surface, it begins to divide. And so it divides into 2 cells, and then 2 cells divide into 4 cells, and 4 into 8, and so forth. And when you come back to the plate, you have the next day, you have a pile of bacteria that you can see with your eye. So
Nathan, you actually have your first question.
Okay, great.
How quickly do bacteria multiply?
Yeah, okay. So bacteria, it can grow from a single cell to a visible colony, which I'm about to tell you what a colony is, but basically it's just a pile of bacteria. That can happen within hours. Like I have some strains of bacteria that they can grow up to colonies that you can see with the naked eye within 6 hours. But typically we'll put a plate in an incubator and let it sit overnight, so 16 to 24 hours, and then we can see these colonies. So send a follow-up question if I didn't answer that, give you enough detail. So what's pictured in right now are several different incubators. The ones that are light gray are, they're all at 37 degrees Celsius, which is human body temperature. And the light gray incubators they're hooked up to carbon dioxide tanks because the bacteria we like to study need CO2 supplementation to grow well. And within each incubator, there's shelves we can put plates on. And there's also a tray with liquid in it that helps humidify the chamber, and this helps the bacteria grow. So when you remove a plate from the incubator, this is what it can look like. So the movie I showed you, this is one of those plates. And these little circles, these are bacterial colonies. And this sample was from a throat swab, and this sample was fairly homogeneous, but sometimes you get a whole wide range of colonies of different shapes, colors, and sizes. And we use this streaking technique to help isolate the bacteria we're interested in studying from a mixed sample. So a throat swab is going to have, could have hundreds to thousands of different types of bacteria in it, and we use this approach to try to purify the bacteria of interest that we want to study. So in the next slide, I'm going to show you a cross-section of a colony so you can appreciate what this is. So some strains of bacteria, they'll start as a single cell, and that cell will continue to divide until we can see this colony on a plate. And you can get 1 billion cells within that colony within 16 to 24 hours, depending on the type of bacteria you use. So here on the left, I have just a half circle illustrating a colony, so the cross section, and I'm showing a scanning electron or a transmission electron microscopy image of just a portion of that colony. You can see it's full of cells. Okay, and so you can just have millions to billions of cells in these colonies. And here is another cross section from region 2 on this colony. And you'll notice the cells are more densely packed at the bottom where they're interfacing with the plate surface. So the inside of a colony is not homogeneous. There can be differences in many different things, including like oxygen gradients. Anyway, I will go on. So sometimes we like to take pictures of colonies, and we have several microscopes in the lab, and this is a picture of a stereo microscope. And if you look down here at the base, we can put a Petri dish there and we can illuminate light through the bottom of the plate. And right here, there's what are called gooseneck lights, and these are lights we can illuminate from the top. So we have the option to illuminate from the top, from the right or left, or both sides, or from the bottom or not from the bottom. We can do lots of different combinations. And I just wanted to share with you some pictures that I've taken on this microscope and some other ones, and that you can look at on my Instagram page if you're interested. So just look up my name or Germ Chaser on Instagram, and you can see.
I have a question.
Okay.
So you were talking about colonies, and what they want to know is, are all the cells in the colony the same bacteria, or do they ever mix together?
Yes, in this example, we believe they are all the same. And bacteria can undergo developmental processes where they actually switch in shape, but they're still the same species. But some species of bacteria associate so tightly with one another that it's hard to purify them away from one another. And so colonies can be a mixture of multiple species of bacteria. But in the images I'm showing you here, we believe they are, it's a clone that has grown up, one cell that's grown up into a colony.
One more question, or do you want to move on?
No, another question's great.
Do colonies ever fight with each other? It's an interesting question.
Yes, they do. And so you can put colonies from different species of bacteria next to one another, or sometimes the same species, and colonies can secrete compounds that inhibit the growth of a neighboring colony or affect how its structure. Okay, so and these types of manifestations that you can see when you grow bacteria on a Petri dish have helped scientists identify antibiotics and other compounds that can inhibit growth. And so if you think about bacteria in the environment, they're really they're experts at trying to defend their turf. And so sometimes they secrete compounds to kill other bacteria so they can claim territory for themselves and dominate or utilize resources in a microenvironment that they want to acquire.
Thank you.
Okay, so I'm going to just show you some different types of colonies, and I find them really pretty, but maybe I'm a bit of a science geek, but you guys can let me know if you like them or not. And I'd love to hear your feedback on what your favorite colony is on my Instagram page. So both of these colonies illustrate a trait called opacity. And so if we look at this colony on the right, you'll notice the growth in the center is quite clear, but then we have several sections of brown growth. So the opacity or translucency of the colony is different. And this can be due to differential expression of proteins from the same species of bacteria, but causing the organism to refract light differently. This colony on the right started out growing as just a plain circular colony, but I left it out at room temperature for like a week, and it started to grow these little protrusions that have different levels of opacity. Different bacteria express different enzymes, and we can drop a little reagent onto colonies. We just drip it on with a pipette, and if the organism has a particular enzyme of interest, it will turn that compound purple. And so some of the bacteria we work with have this type of reaction. And so the colony on the left is at higher magnification than the image on the right, but the image on the right has a mixture of colonies that are positive for this enzyme and negative for this enzyme. So the purple ones are positive. The ones that are white or beige were also exposed to that compound, but they do not turn it purple. Okay, and so this is a mixture. And again, we use that streaking technique to try to purify bacteria of interest away from other contaminants or other bacteria we're not interested in. In this example, I have two colonies that were grown on the same type of media that has a dye in it. And the bacteria are taking the dye up differently than one another. So we have the dye, the plates normally look pink. And so this colony looks very pink, but this one is red. And so this dye is sensitive to pH, and they're interacting with the dye differently. Okay, this is an organism called Serratia marcescens or marcescens. I'm not an expert on this microbe, but it's used in our teaching labs here to teach students about pigments that bacteria can express. And this pigment is a red pigment, and this pigment actually has been found to have anti-cancer properties. And so some of you may wonder, well, why would anybody want to study a pigment, or why would anybody want to study bacteria? We often find interesting things when we study bacteria that have other applications in the health sciences. And so at body temperature, this organism does not express the pigment, but at lower temperatures, it does. And so we're looking at three colonies at the top. They kind of look like peppermint candies to me. And we're looking at a streak plate, the three images on the bottom, just to show you some of the crowding and some of the variation that occurs. So some of the colonies in this streak don't have any pigment and some do. All right.
You have another question, if I could, about how many bacteria do you need in order to be able to see the colony with your naked eye?
It's a good question. I'm not totally sure, but I would think probably 1 million. But that's a good question, and I haven't done an experiment to answer that. So like E. coli that I've worked with when I was getting training during graduate school, a colony that was 16 hours old, I could get 1 billion cells out of. And we have this one strain we work with in the lab now that we're always impressed with. I streak in the morning, and then at 1 or 2 o'clock, I can see colonies on the Petri dish. And I haven't counted how many cells are in those colonies that when they become visible, but I would guess over 1 million. But
Thanks.
Okay, these are cockroaches. Okay, so, and these are, we had a visiting professor from a sister university in Japan, and the Japanese word for cockroach is gokiburi. And this visiting scientist, he studies parasitic nematodes. And so we collected cockroaches while he was here, and we dissected their hindgut and tried to isolate these nematode worms that he was interested in finding for his research. And I plated some of the hindgut material onto some of the plates we use in our lab to grow bacteria, and we got this, an organism that formed colonies like this. And so what we're looking at here is the same colony in both images, but with different types of light illumination. And so the image on the left is with top-down light illumination, and the one on the right is with illumination from below. And so you can see different types of structures depending on how you illuminate the colony. Then I have another example that I wanted to share with you. And this again is the same organism, but we have on the left illumination from the bottom, and on the right illumination from the top only. And when you illuminate only from the top, you get this really weird refraction that looks kind of like rainbow patterns. And there is some literature on this phenomenon, and the bacteria are rod-shaped, and they line up and they align in certain ways that impacts the way light bounces off the colonies. All right, so hopefully you have a better understanding of what colonies are now. I'm going to switch gears a little bit and talk about cellular microbiology. Okay, so I've been talking about bacteria, and that field of study could be referred to as bacteriology or microbiology. And if I'm a cell biologist, maybe I'm studying how the structure of human cells or how they work. And so cellular microbiology is kind of the boundary between microbiology and cell biology, where we're utilizing bacteria in the context of an infection to learn about human biology. How do human cells respond to a bacterial infection? What I'm showing in this slide is a monkey cell. And so I'm circling the size of the cell, and the dots on that cell are Neisseria, and they're binding as diplococci. And this, I just wanted to show you this so you can see the difference in size between bacteria and cells. So typically, a human cell is going to, or a mammalian cell, is going to be like 50 microns in diameter or length, and the bacteria are much smaller. And my lab is really interested in how bacteria interact with human cells or mammalian cells. And so we often culture human cells with bacteria, and this is a picture from my tissue culture room that I share with Dr. Tomo Sugiyama in the biological sciences department. And on the left is what's called a hood, a laminar flow hood, and there's that blue glow light is because there's an ultraviolet light on in the hood. We keep that on for a period of time to help keep the surfaces clean, so UV light does kill bacteria. And we process cells in this hood to try to prevent contamination. Growing human cells in plastic dishes is really, you have to do it in certain ways to prevent contamination. And then on the right here is another CO2 incubator that we grow just our tissue culture cells in, so human cells in this incubator. And so the other incubators I showed you are in my main lab, and we grow bacteria in there or human cells infected with bacteria in those incubators. This is a picture of my student, graduate student Donnie Pickle, and he has a glass pasture pipette inserted into a plastic flask. And he's removing media, a liquid media from the flask with a vacuum. In the back of the hood, you see 2 flasks with liquid, and the human cells he's growing are adhered on the surface of those flasks, and the media on top is feeding those cells. So we propagate human cells in these types of flasks, and then we can collect them and distribute them to other types of dishes that we can then infect with bacteria. And we're interested in studying what happens when we to the human cell during infection. All right, this is from a paper from the lab of Hank Seifert, who's a really respected scientist in my field. And these are images of colonies of Neisseria gonorrhoea. And on the left, we have what are called pileated colonies. And so these bacteria in these colonies are expressing on the bacterial cell hair-like fiber that are called type 4 pili. And these fibers are really cool. They're retractile. And so the fiber can attach to a cell, a human cell that the bacteria is infecting, and they can pull on the infected cell. The colonies here on the right are of colonies that don't have pili, so they're kind of bald. And you'll notice the diameter of the colony is much wider. Okay, so those fibers actually interact with one another, and they actually make these the diameter of these pileated colonies smaller. Now I'm going to show you a scanning electron micrograph of Neisseria gonorrhea infecting a human cell. And so I have a colleague named Dustin Higashi that took this image, and he pseudocolored the bacteria orange. And the meshwork of fibers you see on the bacteria are the type 4 pili. They're kind of matted down in this image because the technique kind of results in that. It's kind of like if you dove into a pool and came out of the water, your hair would be all matted down. It's kind of like that. But normally these type 4 pili are emanating from the bacterial cells and sampling the environment. And if we look up here in the top left corner of this image, there's a little protrusion from the human cell. That's called a microvillus. And the pili from the bacteria can attach or bind to those microvilli. And when the pili retract or pull, that leads to elongation of the microvilli. And so these type 4 pili have multiple functions, some of which are listed here. So they allow these aggregates to form. We call these microcolonies. And they power a form of motility called twitching motility. So when the pili pull, they help move these aggregates across the cell. And I'm not going to talk about these other aspects of it, but the pili do take, I just mentioned DNA uptake. They can acquire DNA from the environment, and this function helps the Neisseria gonorrhea acquire antibiotic resistance, which is a really big problem right now. Okay, any more? Just let me know if you have questions.
Well, there actually is one question, and it's related to what you were talking about. It came in a while ago. So how quickly can bacteria develop resistance? So you just talked about how some bacteria are resistant. So why do they, and over what time scale can that happen?
Right, that's a great question. So they can develop resistance extremely quickly. It's a natural process, and it's spontaneous. So when a bacteria replicates, like during that process of growth from a single cell to a colony, the DNA in the organism, which we call the genome, is copied. And that process is not perfect. And so you can have the mistakes made during that process. And so in a population of 1 billion bacterial cells, some of those cells are going to be mutated at different places in their DNA. And that process can happen within, less than an hour, in minutes. And it's somewhat spontaneous. But if I took like 1 billion bacteria and I put them in a Petri dish that had an antibiotic, most of those bacteria would die. Okay, because they're sensitive to the antibiotic. But a small portion of those bacteria would survive because as they replicated their DNA, they had a mutation in a region that allowed resistance. So it can occur very quickly. And so like when you take a course of antibiotics, it's really important that you maintain, you follow the directions, because if the concentration of the antibiotic drops below a certain toxic level, the bacteria population size can remain high. And when the population number is high, there's more of a risk of developing these spontaneous mutations that will allow resistance to form.
Thank you.
Okay. Okay. We're looking here on the left at a polarized light image of an endocervical epithelial cell. Okay, so this cell line was derived from a cervical biopsy. And this is a site that Neisseria gonorrhea infects in women. And this, the arrowheads are pointing to microcolonies or aggregates of Neisseria gonorrhea. And in the right image, this is the same cell, but we've stained the cell with a fluorescent reagent that lights up green in an immunofluorescence microscope. And we're looking at a human protein. We're seeing where it localizes during infection. So this is after 3 hours of infection in a laboratory dish. And this protein called CD46, which I'll teach you a little bit more about coming up, is clustering at the site of bacterial adhesion. And my lab is really interested in understanding what is the consequence of this. Is this a response of the human cell to infection, defending itself, or is this the bacteria manipulating the human cell for its benefit?
Mm-hmm.
So now on the left, I've illustrated a microcolony. So these are the diplococci of Neisseria gonorrhoea, and the green dots represent that human protein CD46. And I'm going to show you a movie of slices of a microcolony going from the bottom to the top of the bacterial aggregate. And the green in this movie is that human protein CD46. And the blue dots represent the bacterial DNA here in the center. In the corners of this image, we have blue, and those are that's the DNA in the nucleus of the infected human cells. And we have two cells that are partially shown in this movie. So notice this green puncta within the bacterial aggregate. And so we have CD46 that permeates this bacterial aggregate from top to bottom. I'll just show that one more time. And so my lab's really interested in understanding why is this happening. And so now I'm going to go to the next slide here, and I'm going to stop sharing for a moment to give you a break. Hopefully you guys can see my face. Rox, can you confirm that? Okay.
I can.
Thank you. So I'm going to talk about a concept of protective blood. So if you scrape your elbow and you get some dirt in your blood, that's going to have some bacteria in it. There's a system in your blood that immediately can start attacking those bacteria and kill them. And I want to teach you a little bit about this system and how it's one of the defenses the human body has. In my lab, we get blood donations from volunteers, and my student Donnie Pickle lets that blood clot, and then he spins out the clot with centrifugation so that he just isolates the liquid or soluble portion of the blood. That's called serum. And this has a lot of these proteins called complement proteins in it. And I'm going to tell you a little bit more about this complement system that helps protect the human body. So hopefully you guys can see this. It's a vase, but I've put some red cellophane in it to represent blood or serum. And then here's some bacteria. I'm just going to drop those in the blood. Okay. So we can do a simple assay in the lab where we add bacteria to blood, and then we take small aliquots out of this mixture and put it on a Petri dish and grow the bacteria up. And we can do this at 5 minutes, 15 minutes, 30 minutes, and some bacteria are very sensitive to serum and the complement proteins, and they start to die. And so we can monitor bacterial survival over time in response to these proteins in the blood. And my lab is studying this process. So I'm going to share slides again. Here. See if it goes. Maybe I have to escape first. Okay, I think I got it. All right. Sorry for the delay, you guys. All right. All right, so I'm going to talk about this complement system now and this process by which Neisseria gonorrhoea evades complement. So perhaps you watched some debates during this political season, and you'd notice that sometimes politicians evaded the question that was asked. Well, Neisseria gonorrhoea it works to evade some of the immune defenses of our body. And the system I'm going to teach you a little bit about is the complement system. So if the bacteria get past the physical barrier, such as our skin, and get into our bloodstream, this system can attack the bacteria. And there's 3 pathways that play a role, and those are listed at the top here. Don't worry about the details on this slide, but this is a series of more than 40 proteins that can work together to recognize foreign microbes in the blood or other biological fluids in the body. And they converge into the center part here into a protein called C3. And this molecule becomes activated and split, and there's a portion of it called C3b that can attach to a bacterial cell that the complement system's trying to kill. And C3b goes further, it goes on to activate the system a little more, allowing binding of another series of proteins to the target membrane that result in what's called a membrane attack complex. And this forms a pore in the bacterial membrane that causes it to spill its guts and die. Now, this complement system is really tightly regulated. If it's overactivated, it can attack the cells of the human body and cause the membrane attack complex to kill our own cells. And so human cells throughout the human body express regulators of complement. So if C3b binds to a human cell, it can be inactivated. And so that's what I mean on this slide by requires regulation. And I borrowed this or got this slide from my former student, Paul. That's why I have his picture here on this slide. And then this slide I scavenged from my student, Donnie, and he made this little animation that I think simply illustrates how the basics of this complement system. So remember, C3b is one of these central players. We're showing a bacterial cell on the left and a human cell on the right. Now, please note here, Donnie added a scale bar. So the bacteria is much smaller than the human cell. And C3b will try to attach to the bacterial membrane, and that will lead to the formation of that membrane attack complex, and that will kill the cell, okay? Now, if C3b accidentally attaches to a human cell, the human cell can be damaged. And so human cells express regulators of complement that can destroy the activity of C3b and protect the human cell from complement damage. So the cell remains happy, okay? All right, and so I showed you some pictures before about how bacterial Neisseria gonorrhea microcolonies seem to steal CD46. And that's what those green dots are here. And if C3b attaches to the bacterial membrane, CD46 can help inactivate it. So IC3b stands for inactive C3b. And so we think Neisseria gonorrhea is stealing CD46 from the infected cell so it can use its activity to protect itself from complement. Any questions on that?
Well, there is a related question.
Okay.
So is it better for an infectious microbe to be lethal or kind of sublethal, stealthy? So, I mean, you're talking about, you know, a bacteria that is stealing complement so that way it can just sort of hang out. But at that point, is it trying to have a strategy where it's just going to grow really rapidly, or does it just sort of hang out?
So we think the bacteria are doing this so they can persist on mucosal surfaces in the reproductive tract, or in the pharynx, which is another site where gonorrhea infections can occur. And so the first part of your question, in my mind, it's not wise for a pathogen or a bacteria or virus to kill its host because it's basically killing its home. And so I have a little more respect for bacteria that maybe cause chronic infections or cause infections that are asymptomatic. And so they're trying to come to a balance with their host, so they're not killing their host outright, and so they can maintain a stable environment to persist and live in. And Neisseria gonorrhea is really famous for causing asymptomatic infections. So in both men and women, people can be infected but not know that. And these types of infections lead to dissemination of the organism throughout human populations. And in women in particular, it's really can be dangerous because it can cause ectopic pregnancies or pelvic inflammatory disease that can lead to sterility without the person knowing. Okay. All right, so we think Neisseria gonorrhea is stealing this complement regulator called CD46 to protect its own, the bacterial cells from the complement system. And in our lab, we've, so here in this slide, I'm showing a human cell infected with microcolonies of Neisseria gonorrhea, and the cell on the left has CD46 on it, which shown it in green. And you'll see the green dots in the microcolony. So it's productively stealing CD46. And we've made in the cell, we've made a human cell line that lacks CD46. So in this type of infection model, we can infect cells so they can't steal CD46 from the infected cell, and we can then, during the infection, we'll add human serum, which is a source of complement, and we can then monitor the survival of the bacteria under different experimental conditions and see if CD46 is really important for Neisseria gonorrhea survival. And so another thing we'll often mutate is proteins expressed by the bacteria. So here's on the left is our wild type Neisseria gonorrhea that can sequester or steal CD46 from the infected cell. On the right, we have PIL-T mutant strain. So PIL-T is it's the name of a protein. It's for a gene that encodes a protein that powers pilus retraction. And so when you mutate that gene, the bacteria cannot retract pili. And those types of bacteria, they can adhere to cells well, but they can't steal CD46. And we have found in the cell, in our experimental model system, that this strain is very sensitive to serum killing. And we believe that's because it can't steal CD46 during infection. So I'm going to show you another movie now. And what I'm going to be showing you is the processing of samples after we've added bacteria to human cells, let the cells infect for a period of 3 to 5 hours, and then we add human serum, which has complement, and then we start monitoring bacterial survival. And let's see, let's just go to that. And now, if you guys see anything in this movie that you want to ask me about, like a piece of equipment or what I'm doing, let me know. Put it in the chat. So I'm using a glass rod. I've dipped it in alcohol and I flamed it to sterilize it. And now I was cooling the glass rod there because if the rod's too hot, it'll kill the bacteria. Here I'm flaming another glass rod. And then I'm touching it to the lid of the Petri dish to cool the rod. And then I'm placing it on the sterile surface of the agar. Now I'm retrieving a sample, getting a pipette tip, and I'm mixing the sample on an instrument called a piece of equipment called a vortexer. Bacteria will settle over time, so we have to mix the tube so that we're looking at the suspension, so we keep the bacteria suspended. There I put 100 microliters or 0.1 milliliter of bacterial suspension on the plate, and the plate's on a plate spinner, and I spin that, and then I'm pushing the rod back and forth to spread the bacterial suspension on the surface of the plate.
You have a question that just came in.
Okay, great.
What is agar?
Agar, yeah, so agar is something we add to media to help it solidify. Okay, and it's purified from red algae, and it's a very useful tool for making solid growth media in the laboratory. In the past, microbiologists used gelatin purified from like pigs or from proteins from cattle or pigs, and that's just not as useful as agar.
When you grow different types of bacteria, do you have to use different types of growth media and agar?
The agar is usually the same, but yes, there are different types of growth media. So there's many types of growth media, and some help certain types of bacteria grow. You can add human blood or animal blood to media, and sometimes bacteria really like, they need blood to grow. Okay, I'm gonna stop that video, but after I've done plating, I put these plates in an incubator, and then I come back to them in like 48 hours, and this is what you see on the plates. You see all these dots are colonies that have grown up, and we can count these colonies, and that's shown in this picture here. We have an instrument where we have a magnifying glass on it, and light illuminates the colonies, and we use a little marker and a counter to, or a clicker to count the colonies on the plate, and we can do a back calculation to see how many bacteria are present under different experimental conditions. This helps us plot survival ratios or percentages for bacteria. I didn't have time today to talk about some of our other experiments we do in the lab, but Eliza Thapa in my lab is studying a Neisseria mouse colonization model, and so here she's swabbing the oral cavity of a mouse, and she'll take, we dip the swab in some saline, and the mice actually like to lick the salt, I think, off the swab, and so you see that mouse actually holding the shaft of the swab as she rotates it in the mouth, and she takes those swabs, and she's able to quantify the number of bacteria on those swabs using some techniques similar to what I've shown you today. So we do have a lot more time for questions. I do have some more colony pictures I can show you if we have time and we run out of questions. This is one picture of some of the people that have worked in my lab. I'm listing on the left here the students that currently work in my lab and a couple that just recently graduated. Some of my advisors are listed up here. So I got training in many laboratories during my career, and I'm grateful to them for them training me. And we do have some funds that support our research from the National Institutes of Health and the Center for Translational Research, or the unit here at Ohio University. We have people there that have helped us a lot in getting blood donations from volunteers, so we can use the serum from those individuals in our experiments. And again, look me up on Instagram if you want to see some more pictures of colonies. So I'm happy to take more questions and show you some more colony pictures if you want to look at some.
Well, I think that leads into the next question you just got. I like the peppermint candies. What's your favorite bacteria? Well, I've devoted over 2 decades to studying Neisseria species, so it's going to be a Neisseria species. So I do like Neisseria gonorrhea a lot. You can quote me on that. But there's some non-pathogenic Neisseria species that I'm very interested in. And so we and others in the field are discovering new species of Neisseria all the time, and it's a lot of fun. And so I really like some of these bacteria that form really interesting colony morphologies, wrinkled colony morphologies, and different shapes. Yeah. That's cool. Okay, you got a question that goes back to the video that you were showing. What is a vortex and how does it work?
Okay, so in this movie, this instrument here with the blue metal base, both of these are called vortexers, and there's a little, it's just a little machine, and it shakes this pad, and you can when there's a setting on it that's touch sensitive. So if I touch a tube to it, it will cause a little vortex to occur in the liquid. So it kind of looks like a tornado coming down in the liquid in the tube. So we call these instruments or equipment vortexers. So when we're preparing samples, we'll vortex for a minute in this rack on a vortexer, and then we'll hold our tubes in our microfuge tube rack, and we'll occasionally pulse the tube on another vortexer just to make sure the bacteria are suspended before we put them on a plate to spread them out.
We knew this next question was coming, Nathan. Your work is primarily with bacteria, but we are hearing every day on the news about a virus, COVID. Can you explain the difference between bacteria and viruses?
Yeah, okay. So in general, bacteria are going to have a lipid bilayer. So there'll be a membrane that surrounds what's called a cytoplasm, and then they have a genetic material inside that is not surrounded by another lipid bilayer. Viruses don't necessarily have lipid bilayers. Some do and some don't. But viruses, they require a host to replicate. Okay, and so you have viruses of bacteria, and they'll attach to a bacterial cell and inject genetic material into the cell, and they'll induce the replication of viruses. The coronavirus is going to replicate in human cells, and it requires a host to continue to persist within an individual. Okay, there's a lot of bacteria. Some bacteria actually require a host to live in too. So we don't find Neisseria gonorrhea in an environmental reservoir. We only find it in humans. But we can grow Neisseria gonorrhea in a Petri dish. So it has the capability of growing by itself outside of a human host, whereas a virus requires a host to replicate. So that's, if there's other follow-up questions you have on viruses, let me know. I'll do the best I can to answer.
There is another follow-up. What's the size difference? So you said that a cell is typically 50 microns, and then a bacteria is closer to 5, I guess. How about a virus?
So, all right. So, like the bacteria I study are about a half a micron in diameter.
Oh, half a micron. Okay.
Okay. So that would be 500 nanometers. Okay. So a nanometer is 10 to the minus 9th meters. So a virus is going to be even smaller. And so we're dealing with the maybe down to in the range of less than 100 nanometers in diameter, and even smaller. Now there's a new field, fairly new field in the last decade or so. They have been finding giant viruses. And so that's really kind of an exciting area. It's really, sometimes it's easy to find viruses by filtering liquid in the environment. And we often use filters that have a pore size of 0.2 microns. And that will filter out the bacteria, but a lot of viruses will go right through that filter. And so that's a really easy way to purify your sample away from bacteria and just study the viruses that are in it. But they've found now that that pore size actually excludes some large viruses that are being studied now.
Really cool. You know, I have a question. So I think that a lot of people assume that bacteria are all bad. And I was curious, you know, what % of bacteria are bad? Is there really such a thing as a bad bacteria, or is it sort of like where a weed is a plant in the wrong place? You know, are all bacteria inherently good or bad, or some good and bad depending on the circumstance?
Yeah, I guess if we're going to anthropomorphize bacteria, most of them are good. Okay, and we have a lot of bacteria in our guts and in our bodies that actually help us utilize or break down things we eat, so we get more energy from them. And some bacteria in our gut produce vitamins that help us be healthy. And so we're actually healthier because we have a lot of bacteria in us. I think one of the common questions people get, and that we talk about sometimes in my general microbiology classes, how many bacteria are in or on the human body?
Yeah.
I was trying to look that up in case that came up, but there's like 10 to the 13th cells in a human body, and there's about an equal number of bacteria or microbes in the body. And so that not just bacteria, but fungi and other eukaryotic microbes. And it used to be thought that there was 10-fold more bacterial or microbial cells in the human body, but now the more recent data says there's probably about equal numbers. But so when you're walking around, you're not just you, you have trillions of bacteria in you, and it's something fun to think about. So, and most of those are good, and they actually can protect you. So if you think about the bacteria in your oral cavity, when you brush your teeth in the morning, that smooth feeling you get is because you've brushed away a layer of microbes that were growing on your teeth. And the microbes in us, that they aren't causing disease, but sometimes one of the benefits they give us is that they make it harder for bad bacteria to set up shop in us, okay? And so they kind of occupy some of the microenvironments in our body and make it harder for pathogens to colonize or infect us.
Okay. You have a question. Can you tell us more about gonorrhea? What is the disease?
Okay. Yeah, so Neisseria gonorrhea is the name of the bacterial species that causes the disease gonorrhea. And this is a sexually transmitted infection. And there's over 800,000 cases a year typically in the United States. And this organism continues to persist in the environment. One of the reasons it's of concern is it actually facilitates HIV transmission. Okay, and so oftentimes when somebody has a sexually transmitted infection, they're co-colonized with sexually transmitted viruses and bacteria. So another sexually transmitted pathogen you may have heard of that's a bacterium is chlamydia. And so oftentimes people are co-infected with chlamydia and Neisseria gonorrhea and HIV. And the organism, people have tried to make vaccines for Neisseria gonorrhea, and they have failed because the surface of the bacterium can change over time. And so during an infection, the body mounts an immune response, generates antibodies and cells that can attack the bacteria. But those antibodies recognize protein sequences on the surface of the bacteria, but a lot of those dominant proteins on the surface of bacteria or other molecules can change how they look over time. Mm-hmm. Might have an antibody arise that's effective at killing the bacteria, and it goes in and it kills a large portion of the population, but there's a small portion of the population that has changed its surface so that antibody doesn't recognize it. And so those bacteria can continue to persist in the host. And so there's a big effort going on right now in the field trying to identify vaccine antigens that can be used in a multivalent vaccine in the future. Currently the CDC recommends treating Neisseria gonorrhea infections with 2 antibiotics, and but there's resistant strains of Neisseria gonorrhea circulating in human populations resistant to both of those antibiotics, and so we're fearful that we won't be able to treat gonorrhea in the near future, and so scientists are working to identify new antibiotics and develop vaccines to fight this.
So a question, do you ever worry about being infected by the bacteria that you study? Yes, we do. And so so that's why it's very important for us to wear eye protection in particular. Those are probably the most common types of laboratory infections for Neisseria gonorrhea. So accidents do happen in the laboratory, and we try to minimize those by wearing eye protection and lab coats and gloves. But we can work safely with Neisseria gonorrhea in the laboratory, and so I do have undergraduates in my lab that handle the organism. It just takes some training to learn how to handle them safely. We just got a request. They would like to see the slideshow. We have just a minute or two left. If you could show some more pictures and walk us through it.
Okay.
I think you might have a fan club on Instagram now.
When I teach general micro, I try to post new pictures during the spring semester, and so I'll try to get more up there soon. All right, I'm trying to share. Hold on here. Can you see the pictures of microscopes?
Yes.
Okay, these are a couple other stereo microscopes that colleagues have helped me use or loaned to me. And so we take, you see a stack of Petri dishes there, and we take pictures on these microscopes too. Some colonies have really interesting morphologies. And so if you look at these two different colonies, they look kind of like you're looking down on a topographical or a map from a satellite image, and it looks like mountains or rivers. This one is a bacillus species, and this was Dr. Laura Saltman that runs some of the teaching micro teaching labs on campus, and they do an activity where the students swab their backpacks, and this came from a student's backpack, and I spotted a small suspension onto the center of a Petri dish, onto the agar, and so just like 1 or 2 microliters, and this amount of growth took about 4 hours. So you could see it very quickly. This organism is very interesting because the colonies have these radial filaments that extend from the center of the colony. And if you let this grow overnight, it fills the whole plate within 16 hours or maybe even faster than that. So I spotted on this plate like 1 microliter and or 2 microliters, and then overnight it spread across the whole plate. And this organism has flagella, which are like little propellers on bacteria that causes an activity called swarming motility. So the bacteria actually can swim across the surface of this plate much faster than the Neisseria species I studied. So the Neisseria species will never swarm over an entire plate like this. And the image here on the right is just a pseudo-colored or an inverted image, so you can see those radial filaments a little more easily. So it's not normally blue, okay? So the normal color is shown on the left.
Okay, well, I think I need to interrupt at this point. We have hit our time, and I wanted to thank you so much for sharing what is obviously something that you're deeply passionate about. So thank you.
You're welcome. Thank you for your questions.